All right. Hello, everyone. Let me share my screen with the presentation. Welcome to the Town Hall of September. It's been an exciting quarter, actually, even though it has been a summer break. Nope, I know. Even though it has been a summer break, lots of things are still happening. And so we will be very excited to show you. Just a quick reminder of the purpose of ARISE, what we do and what we do not do. So let's start with a general update, a short presentation per team, and the aim is to be finished in 45 minutes so that we have plenty of time also to listen to your feedback or any kind of suggestions or just general discussions that you'd like to join. So please hang around even after the presentations because we really value your input there. But just as a reminder, ARISE does species recognition. Our aim is to be able to recognize all species within the Netherlands using new technology. We have a stream around DNA. So can we retrieve the DNA barcode for every species so that we can also do the meta barcoding on a larger scale? At the other side, we have input for sensors. So digital images, sound, radar, anything that can help us identify species in the field so that we can work towards biodiversity monitoring. That also means a huge AI component, obviously, especially on the digital side, but also on the DNA side. A lot of data will come in, a lot of different data will come in. And so AI is needed to build the necessary components for annotating data, new AI models, and running the models. And obviously, all the way at the backbone is also the data management side of things, which is huge when we talk about the different types of data. This is our team, our larger team and sub-teams that are specialized in particular components. You will listen to Lotte, who is our new team lead for the sampling team. Daniel Kisling is here for the monitoring demonstration sites team. Chantal Huibers for the BioCloud and species reference team. The digital identification side will be handled by Jacob Kamminga, and Kevin Beentjes is our sequencing team lead with all the expertise there. 5 dedicated teams, each with their own assignment, but obviously they have to collaborate to ensure that we deliver the end-to-end solutions for the biologists and ecologists that we are building this for. So what's happening? Besides all the team updates, I always like to give a bit of a glimpse of what's happening, also generally speaking. We had a very nice technical foundation workshop together with SURF and eScience and the Data Competency Center for the Life Sciences. Can't say much about this, except that we try to understand more also across all of the large-scale infrastructures. What are the needs? Do we have common needs? Can we find sort of generic solutions there and work together so that we're not each building different things and duplicate the work? There's also been a citizen science activity in August, but I assume that the sampling team will talk about that, so I'm not going to steal your thunder. And at the same time, we've had a very nice Capgemini collaboration. This was more a big hackathon. So basically, they asked us for a problem. We gave them a nice one. Can you build sound recognition models for crickets and cicadas? So this is really novel because these things hardly exist. Also, the data is very hard to get. So we delivered labeled data, and they actually accrued to over 1,500 participants worldwide through their own Capgemini employees. The results are there. There's a winning team that will also visit us. And we also had the opportunity to announce this cool results during the Climate Week. And that was last week in New York. And they even made an additional VR experience, which is basically a quiz about cicadas, sounds, biodiversity, which we can also host in any given location to engage with the public. So really cool stuff. Save the dates, because there's a lot of things going to happen in the very near future. 1, not this Saturday, but next week Saturday, there's a barcoding NL event. So if you don't have anything to do in that afternoon, join us, because they will talk more in depth about what it means to sample to retrieve this species reference database that we're all hoping to get for the research community. Every species, we have a specimen and a barcode. and we need the help of the community to get there so that we can give it back to the community to do all sorts of research. Second date, please mark in your agendas the next ARISE Day, which is already planned on the 27th of March. We don't have a program yet. If you have ideas about what we should be doing that day, do let us know. So we still need to decide on keynote speakers and the actual program, so any input is welcome. Then, just a quick reminder, this is what we're talking about, for instance, around the barcoding NL event, but everything around ARISE basically revolves around this. Can we build this reference database for each species? So let's start with those use cases, right? Can we go from collecting information in the field and optimize as much as possible, or scale up as much as possible the identification of species from sample or specimen in the collection all the way to exploring the data on an interactive platform? And the same goes for the digital sensors, where we do the same, and both of those streams will need to come together on the ARISE platform, which exists, but the services will be developed through time. Then I'm going to hand over. Lotte, all yours.
Yeah, that's me. Hi, I'm Lotte. I joined the sampling team the 1st of August, so I just started as the new team lead for the team, and I will give you our latest updates today. Next slide. Yeah, so this is our team. We are now really up and running. We have quite some sample managers, and I am, of course, the new team lead. But what is very nice is that we will also have space for some new volunteers from January onwards. So the volunteers work with us in the lab on all kinds of tasks. And yeah, also we see what their preferences are. So if you'd like to learn more about that, or you know someone that might be interested, so please feel free to contact me. So a short recap, what do we do? The sampling team is in charge of filling the target list, which lists, of course, all Dutch species that we still need DNA from. So we do that by sampling in the field and also in the collection. And we include metadata about specimens, for instance, the collection location or the date. And then after DNA extraction, we store all the voucher specimens in the biobank collection. And as you see in the picture on the right, we now use new plates. They are on the left in the picture. And this allows us to store 800 instead of 200 specimens while using the same space. So that's really nice. Okay, next one. So some numbers. We need around 20K barcodes in total. So for that, we estimated that we need to sample 350,000 samples to fill all the gaps. So we want to go from 5K to 50K samples per year. And this year, up till now, we sampled 10K specimens, and we hope to achieve 20K by the end of this year. So that results in 30K samples in total since the beginning of the sampling team. And very exciting, we are now curating the first DNA data in close collaboration with the sequencing team. So Gavin will tell you a little bit more about that. Okay. So with these numbers in mind, you can imagine that we want to standardize as much as possible for all of our processes. So for the actual sampling, or the team actually created these sampling kits that are now fresh and new on our desks. Now we first have them for the insects. And this makes it a lot easier for us to provide you with everything you need to sample for us. So there are tubes in there, registration numbers, there's a manual. And in the manual, there's also a link to the online sampling tool, which we use. You see it on the right for collecting all the metadata. And there will be a new version of this tool probably next week. So if you want to know more about sampling for us, please contact our collection manager, Tim. And I will also give a demo about the new sampling tool, the online tool. So if you want to be invited for that, please send me an email and I will invite you. Yeah, I thought I expected some images there. So yeah, we had a really nice field day in the Biesbos in August. We visited the summer camp of the youth nature organization JNM. So we explained a little bit about what we aim to achieve as ARISE and in collaboration with BGE. And they went into the field armed with the online sampling tool and with our target list. And we actually went home with some new species for the target list. So we were very happy. Okay. You can pull up the next slide. Yeah. So besides collecting in the field, we use collection mining also to fill the target list, and we collaborate with Biodiversity Genomics Europe for that. And Astrid, our team member, has been very busy taking photographs last month. She does that still now one by one. So you see it on the left, picture of the specimen, picture of the labels. But next we want to implement a grid camera system, and that can photograph an entire drawer in about 30 minutes. So this will also, of course, helps us a lot, make it everything photographing and imaging way faster. Yeah, next one. A little bit more on collection mining. We mentioned it during the last town hall. We are now almost finished with the parasite collection that was donated to Naturalis by Herman Kremers. So this is 1,800 specimens. They were preserved in ethanol and glycerine. And yeah, on the right you see how we subsampled it in the shallow plates. And of course, we also want to take high quality images of every specimen in the plate. So we have a digital voucher specimen. Yeah, so we are very happy that our new tool, the Keyens microscope, arrived. So you see an image video on the right that I took during the demo they gave when they installed it in our lab. So this can take 96 high quality stacked pictures of a plate in less than 15 minutes. So this will help us, of course, a lot in our workflow. So what's next? Collection mining. The first BGE samples will go to the lab, mainly pollinators. And we will implement this grid camera system to photograph drawers. And the target list will be hopefully updated with new DNA results that we are now working on. So hopefully next time I'm able to give you some numbers on that. Yeah, because we take a lot of images, we are also working on proper storage for that, because it's such a large numbers. And yeah, of course, barcoding NL, Elaine already mentioned it. So it's next week, and hopefully a lot of experts and collectors that join us on that day will help us in our efforts to collect all the species of the Netherlands.
Thanks a lot, Lotte. Excellent. We move to Kevin.
Yeah, that's me. So yeah, I mean, I can introduce myself, but Elaine already did that. So just as a reminder for the sequencing team, what our main goal is to get from DNA extractions to clean sequence data, with the emphasis on clean sequence data. And today I thought I'd present you with some numbers of how many specimens we've already processed. So within Naturalis, we're working mainly on fauna, and most of that is insects, actually. So these come from both material provided by collectors, which Lotte mentioned, which is identified specimens, but we also have a lot of unidentified specimens that come from Malaise traps. So far, we've almost processed 20,000 specimens for DNA extraction. I think some of my technicians are doing DNA extractions in the lab today, so we might have already hit that milestone today. For about 18,000 of them, we've done PCR. So we've amplified the barcode fragment, which was successful in about 87%. So that's quite a high percentage. Just so you know, 100% success we almost never get that. And of those specimens, we now have about 16,800 sequences from 13,400 specimens. And so you see there are some specimens where we get more than one sequence. And so we're also, of course, now with our new sequencing technology, finding all these like additional sequences from specimens, from parasites or contaminations. But yeah, so we're now in the process of validating and identifying some of these sequences, which in part is still a bit of a manual process. And as you can imagine, for about 17,000 sequences, that might take a while. And so we're already sitting together with the bioinformaticians to also see if we can write some smart scripts for this to make our work a little easier so that we can give some feedback to the people that gave us specimens. As you can see on the next slide, there were some interesting results. So in about more than 200 specimens, we found DNA from an eel. And in about 170 specimens, we found this caddisfly, which is a Dutch species. But when we look further into these identifications, we found actually, what you can see on the next slide is that those were contaminations. So that eel was actually a bacterial sequence that someone put on both, stating it was an eel, and that caddisfly was actually a human sequence. So this sort of already highlights why we are building this Dutch reference, because we need a validated reference where all these sort of misidentified contaminants are excluded, and we don't run into this issue where we find eel in every sample that we process at one point. Similar work's being done at the Westerdijk Institute, where basically there's two different projects. First of all, there's the sequencing of soil fungi from their collection. And the idea is to have taxonomically validated names for all the Dutch soil fungi in the collection. But of course, to do this, there needs to be a lot of comparison and a lot of molecular data need to be obtained from all basically soil-related fungi genera in the collection. And so now there's about 1,500 soil fungi being worked on, but in the larger scale, there's about 7,500 strains that are being processed. Of course, here also there's a lot of work to do validation. So they're actually doing phylogenetic analysis, seeing that what we also see with CO1 for insects, sometimes one marker is not enough. And so there's a lot of secondary or even extra markers being sequenced. And then the other project running at Westerdijk is actually sequencing cultures that have been obtained from soil samples. So there's a variety of projects running, including citizen science projects where soil samples have been collected by school children. And from these soil samples, different fungal strains will be cultured. Of course, this only works for strains that can be cultured. And then hopefully using the molecular data, these strains can be identified. In the next slide, yeah, you can see some of the numbers for this project. So there's about 9,000 strains already cultured. And most of them have already been sequenced for 2 barcodes. But there's also a whole range of them that need a lot of extra work being done. With all these different barcode markers to be able to distinguish closely related species. The next slide has an overview of one of those projects. So here you see what we call a so-called Corona chart, in which basically you can see the whole diversity of one of these projects shown a couple slides ago. The pie chart on the right shows basically all 9,000 strains that have been processed in these projects. And there you can see there's about 10% so about 850 strains that are qualitatively new. So these are strains, not species. So there might be some strains that are the same species, but these might be species that are new, in some cases, to science. So that's some of the numbers that we've been working with. Of course, I can't let this town hall go without showing some of the fancy new equipment in the lab. So here you see the new extraction robot that Westerdijk has recently bought, so that it can also ramp up the production a bit more. And of course, we finally, in the next slide, have our baby up and running. So all the training and programming has now been done for our extraction robot as well. And so hopefully it can start crunching out DNA extracts very, very soon. And that's basically it for me for today. Oh no, of course we want to look to the rest of the year. So for the data analysis, we're still working on some final bits of the pipelines, so bioinformatics, which are almost fully automated right now. Of course, the whole validation needs to be a bit improved because that's now a lot of manual work. And then hopefully soon also the core sequence cloud, so the central storage of sequence data, will start taking shape. And we've also been working with some meta barcoding proposals for projects, which I've talked about in one of the previous showcases or town halls. We now have some interesting projects that we selected, and we'll start doing some meta barcoding pilots as well soon.
All right, thanks a lot, Kevin. Just a few disclaimers. So a lot of the work that is now happening within the sampling and sequencing team is actually in collaboration with our European sister program, Biodiversity Genomics Europe. So BGE, you'll see as an acronym coming flying past. This includes the citizen science, this includes the sampling protocols, and this includes the sequencing. So that's all happening in synergy. Another little disclaimer is, as you've noticed, we're really getting into the final step of end-to-end, where we say we can now have the sequences all the way connected, but we still need to validate, and that's still a manual step. And this is also the reason why some of you that are out there as experts that have been, gracefully giving us the samples have not yet sometimes given the information out of the machines back to you, because we're still in the process of validating some of those steps to make sure that we have the appropriate sequence, but also to hear back from you how you want to look into your result basically from your sample. So if you're out there, please, allow us a bit more time to get that final bit of the puzzle solved. And hopefully we see you at Barcoding NL as well. And then the very last disclaimer, I would say, you also notice that the end-to-end is pretty ready for those researchers out there that want to make use of the nanopore workflow that has been developed through ARISE and should now become available to you. If you have interest there, please connect to our head of the lab at Naturalis, Elsa Duim, and we can see what we can do. No promises yet. I'm saying we're getting ready for this, but that's the appropriate way to try and get it to work for your project. All right, let's hand over to the field side around sensors. Daniel, please go ahead.
Thank you very much, Line. So my name is Daniel Kissling. I'm from the University of Amsterdam, and I'm leading the team on monitoring demonstration sites. We are, besides myself in the team, we have Rotem Silber, who's basically a project assistant project manager. She helps with setting up the field sites, the PR work, the relation to our stakeholders. We have Lindy Schneider, who is a monitoring technician. She helps us with setting up the sensors and maintaining them. And we have Julian Evans, who's the head behind all the data management that we are doing. Next slide. So the focus of our team is basically on digital sensors in the field. We want to innovate biodiversity monitoring with digital sensors. And you see a couple of those sensors that we have been deploying, from wildlife and insect cameras to acoustic devices, radar, aerial cameras, and Nestbox cameras. Next one. We have also worked at a number of sites. You should see on the next slide, basically the overview. We have 3 main demonstration sites. Thank you, that you see with the black dots. This is the main sites. No, just one back, please. So these are the main sites that we're working at. This is Oostvaardersplassen. It's a wetland and grassland nature Natura 2000 site. We have the Amsterdam Savate lying down, and that's a dune ecosystem at the coast. And we have Amsterdam City mostly at Artis Zoo as an urban green space. But we also have a range of other sites that you see in red, where we're doing deployment of individual sensors and testing sensors in different environmental conditions. Next one. So what I would like to show today and focus the presentation, my part today, is one particular use case that we have been developing in the Oostvaardersplassen and on Terschelling. And we focus there on the automated monitoring of geese, where we're using wildlife cameras, basically to understand the role of geese and the dynamics of geese, the seasonal and daily temporal dynamics of geese in those ecosystems. They are important grazers, so they take out nutrients, but they also bring nutrients back, and they are quite mobile, come in and out of those systems. So we want to understand what are the dynamics. Next one. So I want to show with these examples what we have been doing to answer these questions. We have at those 2 sites installed 10 cameras at Oostvaardersplassen in wet and dry grasslands, and 8 cameras in the polder of Terschelling. They're all facing north so that we don't get problems with the sunshine in the images, and we have a standardized design. Next one. So the cameras are Sniper Commander 4G cameras. These are cameras that can automatically transmit the images via 4G. We have used wide lenses, which gives us a broader picture on the landscape. On the right hand, you see our setup. We have installed them on poles about 2 to 250 meter height. In the Oostvaardersplassen, they had to be protected against horses and cows. That's a rewilding site with large herbivores. And on the right hand, you see the tall poles that we have been using at the polder. So the 4 data transfer allows us to automatically transmit the data, and we also operate the cameras with a solar panel so that we don't have to exchange the batteries all the time. And we use a time-lapse function, so not using an infrared sensor, but time-lapse, which allows us to get continuously images every 30 minutes. Next one. So here are 2 example pictures of geese. On the left-hand side, you see that there are huge flocks, but also a lot of the individuals are far in the back. And on the right-hand, you see empty images that we're taking, because we're taking it every 30 minutes. But that's also really important, because we want to measure the absence of those species also at those sites. Next one. So what we've been building is basically what Elaine also emphasized already, an end-to-end pipeline. Can you go one back, please? And this starts with basically the automated monitoring, where we have the 4G data transfer and the cameras in the field that I showed already. And we are transmitting those images automatically to our server, to the monitoring demonstration site server, which is in the University of Amsterdam. And that allows us on our portal, on our server, to do 2 things, at least, which helps us to monitor the equipment. Next slide. And this is on the one hand that we can, from our server, have a near real-time view of the images. So we see what the cameras actually are recording in the field, whether they are operating. And on the lower bottom, you also see that we can monitor the sensor performance. So the blue bars basically show how many files are recorded, and we can measure over time whether the camera is still operating, how much is transmitted. And on the right-hand side, the red curve shows the measures of temperature as an indication of the field conditions. So we can monitor the equipment also in the, remotely from our server. Next one. So from the MDS server, we have established by now a connection to the BioCloud system. You will hear more about this later. About the BioCloud, we can import our images to the BioCloud with an API, and then move them to the DSI, the Digital Species Identification Platform, that you will also hear a little bit more later. And the data are there, can become available there, and then we can basically start with an AI algorithm for automatically counting and measuring the geese. Now, to get this established, next slide, we have worked with a student project from the Hogeschool van Amsterdam. So they are basically AI students that can help to develop algorithms. And they have a project where they used a number of algorithms, among others, the YOLO version 8 algorithm for object detection and a number of annotation data, and have basically developed this algorithm for counting geese. And this, one of the big advantages is that they already started to do this in the algorithm schema that the DSI is using. So we can put this in the DSI platform. Next one, we also worked on basically getting more annotations. So basically annotating a lot of the images with my research group at the University of Amsterdam, where we then basically labeled a lot of these images to be fed into the algorithm. Next one. So I think the next slide shows you some examples, or to one example, for instance, here the raw image on the top and the algorithm as it is in its current state in the detections. Below you see a few numbers of the precision and recall. You can see that this is not yet a perfect algorithm, but that it allows to detect a lot of the geese. There are some issues with it. A number of individuals are not yet well detected. And you can especially see that in the far back, it's very difficult to at this point that the algorithm detects actually the individual geese. So there's more work to be done for the algorithm development. But the nice thing is that we have this algorithm now also available in the DSI algorithm repository, and that we can, with the whole pipeline, basically start a massive processing of tons of images to basically count the geese on a temporal sequence over time in many, many sites. And on the right bottom, the blue bars show just one example of analyzing one camera over 3 days. Even with the algorithm that we have right now, we can already nicely detect the dynamics of geese populations at the sites. So we want to basically deploy this then with a whole data set in the next couple of months. Next slide. So the next step are basically to improve the algorithm. For this particular example, we need to do more quality checking of annotations. We want to annotate more data. We also want to use more AI-assisted annotation, that we can focus our annotations on those images and things where we need, where we have the biggest need, and we then can retrain the AI models to get better detections with larger training data sets. On the other hand, we have also been working with our UvA Technology Center to develop a new camera. We are basically prototyping this camera now in one of our sites. It's a Raspberry Pi wildlife camera that allows us to also remotely connect to the camera, but also to get much higher resolution images, which may allow us in a better way to detect also the geese that are in the far distance. So this is the example of that we're starting to get these end-to-end pipelines with one example that I wanted to show you today. Thank you very much.
Okay, good morning. Thanks, Daniel. You nicely sneak previewed the DSI stuff, right? So this is a great connection. My name is Jacob Kamminga. I work at the University of Twente, and I'm the team lead for the DSI, or the Digital Species Identification team. It comprises software developers and researchers from the University of Twente and Naturalis. So if you're a researcher and you have collected data before, I think we all know this this feeling. At some, especially if you use a lot of sensors, at some point you have this huge pile of data which you need to plow through. Next slide, please. So in the end, this is what we want, right? So we expect a machine that helps us to go from heterogeneous unlabeled data to labeled and organized data, so we can do our analysis on this. So this basically boils down to 3 main steps. And I want to talk a little bit about this, not to just tell you what we're doing and what we're going to do, but also to give you a little bit of idea why we are doing this and how we are making really good progress at the moment. So first, you want to organize this huge amount of data by tagging and filtering, which I'll show you in a minute. Then you want to gain biodiversity insights by detecting, identifying and annotating species. This can be manual, but of course we want this to be automatic, and eventually provide structured output. Algorithms, they help automate these processes, right? And over time they become better to perform increasingly complex tasks. So this looks a little bit like this on the next slide. If you've been here before, you've seen these slides. Just a few examples of algorithms that I'm talking about. So we want to do this on sound using audio classifiers. Oh, I'm not sure what Elaine is doing, but now we're four slides ahead. Can you go back to the sound slide? Yeah. So we want to use algorithms to process audio, detecting, for example, bird species. We want to use algorithms to detect species on images. On the next slide, for example, birds, but perhaps also their prey. So this is a huge scala of things that we aim to address. Next slide. We've been focusing a little bit on Diopsis as a use case. So this is an insect camera. So the next slide, you'll see data from the Diopsis project, where the goal is to detect and identify insects on the Diopsis insect camera. So talking about organizing and filtering your data, when people talk about AI, what has been done, people don't really talk about, is data cleaning, right? So if you deploy a sensor in the real world, you have to deal with real world data. So a lot of unusual stuff happens. Cameras break down, data transmissions fail, plants start growing around your sensor, the sensor is being maintenanced, agoutis show up if you're in Panama. All sorts of things happen, and these actually have an effect on your analysis, right? If you, for example, have 2 days of black images and the algorithm doesn't detect anything, you may think, oh, there are no insects, but this is actually wrong. So you need to filter black images. This is maybe a very dumb example, but it's a good one to show you what we're doing. So what we've done is added the first steps towards tagging and annotating. So here you see the actual output of a camera. And there's some black images, some corrupted images. So if you go to the next slide, you can now add a label to these images. So here I'm adding a label black image. And on the next slide, on the top here in our system, you see you can actually add filters. And we are increasingly adding more and more filters to help you organize your data. So here I filtered by a sensor, and now I'm going to filter by excluding the label that I just added, which is black image. So on the next slide, you'll see that now the black images are gone. Very simple example. Now you may think, okay, you expect me to go through my millions of images to tag them myself? No, but initially someone has to, right? In the first step, which we can then use to learn and train an algorithm to do that for you. So this is an example of how this is an iterative cycle. So on the next slide, this is what we're going for, a continuous process where algorithms over time improve, enhance, and actually help you solve more and more tasks. So initially it may find just geese. You want to filter black images. So you filter your data based on the algorithm where it didn't find geese. It may have missed some, so you correct it. Maybe you find something new. And then the next cycle, it will actually filter your data better. I'm just randomly using the geese example because Daniel was nicely showing this just now. So short update, we are adding algorithms as we go. So now we have Diopsis available and an algorithm called Megadetector. This is it seems pretty simple, but it actually detects animals, people, and vehicles and camera images, which can be used in this kind of workflow that I just showed you, right? So if you want to not have those things in your data set that you're analyzing, you can already run this. And this is for a proof of concept to get into this kind of workflow. Like Daniel said, the geese counting algorithm is being added. It should be available also soon to use on the actual camera trap data from the MDS team. We're nearly done with another human detector, which is also a student project, like the geese counting algorithm. And Dan Thowell and 2 developers from Naturalis will spend time to add a new bird classifier for sound analysis. And the next slide, some updates on the media browser that I just showed you. we are currently steadily handling 1.6 million media items. And so this is what we've been working on mostly, making this work robust and as swift as possible. But the data filters I just talked about, you can now filter on a sensor, on dates, media types, labels that you can now add yourself, and also data sets that you have created. We call this dynamic filter because we are adding filters as you may want them, and as we think they really increase your workflow, improve your workflow. So planning for the next quartile, we are preparing to handle over 50 million items together with the BioCloud team. And filters we're adding, so for example, being able to filter on what your algorithm has said about your data, and also the number of detections. So if I go back to the geese, if you want to only look at images where there were no detections to see if something was wrong, then you can add a filter to say, I want to see all images which have no detections, and then you may quickly find the ones that were missed by the algorithm. And filtering by location is something we heard yesterday during our first feedback session. And on the bottom, show and filter concurrent algorithm predictions. This means that if you have one item but multiple algorithms which said something about that media item, like for example an image, then we are going to work on being able to filter what you see on the image. So for example, multiple bounding boxes at the same time. Next slide. Yeah, so like I just said, yesterday we had a pretty huge milestone. We had our first end user test at Naturalis, which is great. We also had room for the day, and people used our actual system that is now online. And yeah, overall, everyone was very excited, and we got lots of great feedback. So our focus for the next quartile, I already mentioned some of these. We're going to enhance the media browser, visualize and filter the predictions. We're going to integrate with external data annotation tools. Maybe I should add CFET here. I was just thinking about it when I saw Daniel's presentation. But we have a student in Brabant, Fontes Hogeschool, who is going to integrate Label Studio with our platform. We are starting to integrate Intel Gati, which is an advanced annotation platform, which trains algorithms while you annotate. To really speed up the annotation part. We have been working on user management together with BioCloud, which we really want to finalize this year, so that you can actually create a user and organize projects, etc. And yeah, the goose counting and bird sound classifier should be available this year. That's it for DSI. Thanks.
All right. I think it's me up next. So Chantal Huibers for the BioCloud team. Our team is working on all the stuff underwater, I always say. What you can see is the tip of the iceberg, which is the fancy stuff shown on a platform or a website. But underneath that is a lot of code that the developers are putting together in pipelines to actually make these services available end to end. Next slide. So one of the main things that we're doing is building this very large data lake house. I hope one day I'll sit on the deck of the lake house, look out over the lake and enjoy the view. And in the meantime, you can enjoy the actual data that is in the lakehouse. So I'm not going to go through this in detail, but it mostly means that we are building a data architecture that can handle input from various data sources, whether that's images or DNA data or sound recordings. We ingest that all and then put the metadata in our data lakehouse, where we follow a few steps in actually enhancing and transforming that data, harmonizing it so that we can actually use filters like give me all the data that we have for a particular location, regardless whether that's an image or a sound or a DNA barcode. And at the same time, we're also processing all that, all that payload or blob data, so all the images, the actual files of an image or a sound. Well, as you just saw, we've got 50 million Diopsis camera data. We have, I don't know how many, 20,000 DNA sequences to handle. So there's a lot of data there, and then we push that through to the other side, so you can actually view that data in portals like the ones that the DSI team is building, or other things to come in the future. So for now, we have the pipeline ready for both the Diopsis insect images and the wildlife camera images that we get for MDS. And one of the main focuses of the team at the moment is migrating our data pipelines to Databricks. You might have heard this before. Why are we doing this? Version 1 of these data pipelines were built with a tool called Spark. Very powerful for data analysis, very scalable, very useful for what we want to do, also very complex. I think the team has spent months on actually making it work, configuring it. And what Databricks does is actually a tool on top of Spark. So it uses all the powerful features that we need of Spark, but out of the box. No complicated deployment processes, fewer tools that we need to master. We have a relatively small team, so this is really going to help us ramp up, adding more use cases to the whole data pipeline. So we're in the process of putting what we already have, the code, onto Databricks, so it can be used there. Next one. One of the other main things that is being worked on at the moment is the implementation of persistent identifiers. And I kind of want to spend a little bit more time on what that is. So to make all the data findable and accessible and interoperable and reusable or fair, everything needs to have a unique identifier that is persistent. So it's never going away. Like your personal identification number that you have stamped in your passport. And a PID is linked to a whole bunch of other information about an object. So I'm sure most of you are familiar with things like a DOI. Our ARISE abstract that we once put in has a DOI, and the DOI links to, for example, who wrote this paper or abstract, what's the title, where was it published. Another example of a PID is a org ID. So here you see my org ID. It's persistent for me. And if you go to the org ID website and you put it in, you can see where I'm employed, where I was employed in the past, what kind of publications I have. So we also want persistent identifiers for all the data objects in ARISE. Next slide. And for now, we have kind of identified the steps, what we need to do this. So we generate PIDs in the data pipeline at the step of the enrichment process of the data. So we check whether data that comes in already exists. If not, hey, this is new, we need a PID. We actually use the PID service at SURF to mint and handle all these PIDs. A PID exists of 2 bits. One is what we call the prefix. You see the line there in green. This will be the same for all ARISE objects. So you can always identify, hey, this is a data object from ARISE. And then the bit that's behind that is actually unique for every data object. So we also look at what metadata do we need for every PID, and then we store it in the BioCloud. So anyone who has the PID can actually resolve that and see the information about that data object. What have we done so far? So we've built the connection with the SURF PID test server, and we've implemented our BioCloud handle service. So we can actually now mint a PID, look that up, and the next step is to fully integrate that in our BioCloud pipeline. So pretty good work in progress. We do this also in collaboration with the people from Disco, because they already started doing this before us on the digital specimens, and they already sorted out a lot of the things we needed to know. So we're building on top of their knowledge. Yeah. And then one of the other things we've been looking at is authentication and authorization. So when you want to use data that is in ARISE, how do we make sure that users, that we know who you are, and that we know what you can actually access or not. So not everything will be public. We are working towards as open as possible, but we're also an infrastructure that facilitates research that might still be in the process of actually analyzing data before it can be published. So we want to give users also the possibility to have it private while they need that. So at the moment, we have a production environment up and running for logging in, and we've actually gone for a hybrid solution here where we use both Keycloak and the Surf SRAM tool. So through Surf, they are connected with over 1,000 institutions in Europe, which means that you can log in with your own university credentials. So no new ARISE account, but Keycloak was actually much better in handling groups and roles and rights. So we've put in a hybrid solution, which is now actually ready to be implemented in the tools that we already have. And then next is, once you're logged in, what kind of data can you actually access? That's something that we implement in Databricks in the pipeline, and that is now in progress. So hopefully by the end of the year, we can provide all the users with a login, or well, they can use their own credentials to log in and then only see the data that is either their own or publicly shared. Yeah. We've done quite a bit of work that you cannot see, that I cannot even see. I don't really want to go into details, but if you think, well, what are these people doing? We deploy or we develop CI/CD pipelines, so it's much easier to test when we build new stuff, whether something doesn't break. We need logging tools to actually see what's happening at every step. We have a Python library that contains all the shared code that we use between different repositories. So there's a lot of work from the development team to make the whole system work much smoother. And then the next one. All right, so our outlook from the backend side for the next quarter is really focused on the Databricks migration. Our big milestone, hopefully this year, will be to have this connected to DSI so that they access the data not via our initial pipeline, but through Databricks, as well as have all the diopsys and all the MDS images available behind a user account so that people can only really see what is shared with them. We're also working on incremental ingestion. So at the moment, we're just dumping all the data of the last years in our system. But once we have that, we just want to ingest new data that comes in every day. So that's something that is being implemented at the moment. And I already talked about PID and authorization. One last thing I want to show on the next slide. So while the developers are super busy working on everything underneath the water or behind the scenes, I've also been discussing with the team leads and one of our UX designers that we hire externally about, so when a user comes to the ARISE platform, what will they do there? We call this our user flows. So people come to us because they want to hand in a sample, they want to look up a barcode, maybe they have a sensor. There's a lot of complexity in the different ways a user can enter the system or go through the system. And we now have a way more detailed overview of everything that we think a user might want to do on our platform. This will really help us to decide which services and applications we still need to build and prioritize how we're going to do that. So hopefully you'll see a little bit more of what this means for the UI, the front end of ARISE in the next town hall. So that was it from me.
So thanks everyone. A little bit more than the 45 minutes, but that's also my fault for the technicalities. Thanks a lot for joining. I'm going to stop the recording, but please hang around if you would like to join some of our discussions. So hang in there. Just going to see if I can stop this and stop the recording quickly.